So, our learning unit on lipid metabolism probably represents the largest kind of component for the next set of chapters that we'll have for an exam, which is going to be on lipid metabolism, protein metabolism, and then nucleic acid metabolism. So again, this video is going to be the longest because this represents probably about 50% of the content for the next exam. So again, we've been very carbohydrate focused in our understanding of kind of fuel catabolism. And a lot of what we've learned already in terms of citric acid cycle, electron transport, oxidative phosphorylation is going to play in here. But what we're going to move into now is understanding sort of the second fuel molecule that we really have. You know, glucose and carbohydrates being sort of the first class that we learned all the pathways for metabolism. And now we're going to understand lipid metabolism. And again, once we get down to acetyl-CoA, for example, then we don't need to learn anything new necessarily in terms of fuel breakdown, because that takes over at that point of citric acid cycle and it kind of becomes a common metabolic pathway. So again, we're going to have several lectures here on fat metabolism to kind of pull all of this together. One of the things that we first have to consider is the fact that we're going to have both exogenous and endogenous sources of lipids. So things that we'll consume in our diet, as well as things that are kind of present already in our body, specifically adipose tissue. So we will spend a little bit of time talking about digestion, absorption, and transport of lipids, importantly because these are a new class of molecules and that they're hydrophobic. So we need to think about transport issues. We'll also revisit ketone bodies as they relate to a water-soluble but endogenously derived lipid fuel. So conceptually, again, we mentioned already acetyl-CoA is an important branch point metabolite. So we'll kind of talk about how we get to acetyl-CoA from the metabolism of lipids. And then again, once we get to acetyl-CoA, depending on whether we are in a fed state or neat state, determines what happens with that important branch point metabolite. Again, highlighting the importance of understanding how these different hydrophobic substances are transported is going to be key in this learning unit. So in our first lecture here, we're basically going to talk about exogenous lipid metabolism or digestion as it occurs in the stomach, small intestine, how we get molecules to the bloodstream, and then again, delivery to three important tissues that we'll focus on in this chapter, which are the liver, skeletal muscle, and then adipose tissue. So first slide here was really just a review, kind of conceptually remembering structures of carbohydrates from what we learned in Chapter 8, and then structures of lipids. Remember, lipids really is a catch-all for anything that's not water-soluble, but we're really focusing here on two main sort of kinds of lipids. The main one here that we're talking about from a dietary standpoint are triglycerides, which are fatty acids esterified to a glycerol backbone. And then we'll also spend some time talking about cholesterol in this learning unit. So just again, reviewing a few key things here, you do have a homework question that allows you to sort of summarize some of these things in sort of a table format. I always want you to remember and be able to know the structure for beta-D-glucose, nature's important hydrophilic circulating fuel. It's important to maintain blood glucose at 5 millimolar concentration. And in previous chapters, we've talked about the top-down and bottom-up approaches to remedy low blood sugar. Again, the digestion or the degradation of glycogen in liver and skeletal muscle via glycogenolysis to generate glucose from that top-down approach, or the synthesis of glucose from non-carbohydrate precursors in gluconeogenesis. So the reserves that we have for carbohydrates are fairly limited, and glycogen is really the reserve of glycogen, and that lasts only about 12 hours. Importantly, still remembering kind of how much these fuels are worth from a caloric standpoint. So carbohydrates are worth the same as proteins, which is 4 calories per gram. So again, just a real quick review there of the salient points for carbohydrates and specifically glucose. So again, if we come over here to our right side here and we think about triglycerides, right? So triglycerides really represent a glycerol backbone that is esterified to 3 fatty acids. Some important things to think about with these fatty acids. I'm fine if we draw them looking like they are trans because that's way easier to draw. But remembering that we have the cis configuration for almost all of our naturally occurring fatty acids. When we talk about how many carbon atoms we have there, most of the naturally occurring fatty acids that we will encounter will have even numbers of carbon atoms. When we talk about them being saturated, that's what we kind of have shown up here, a saturated fatty acid. Unsaturated fatty acids, when we introduce double bonds, have a few specific rules. Again, we're going to have these double bonds never in conjugation, never before carbon number 9. And then we have some specific nomenclature for omega-6 and omega-3 fatty acids, and that refers to the position of that double bond from the end of the fatty acid side chain, or the fatty acid chain that we have there. Again, if glucose is nature's favorite hydrophilic circulating fuel, the hydrophobic storage fuel that is preferred for by nature in your bodies is triglycerides, and that's the main storage molecule in adipose tissue. A couple of other things that we're going to sort of talk about, and they become relevant later, is this refers to small chain, medium chain, and long chain fatty acids. So again, small chain, we discussed this back in chapter 9. Small chain is really anything less than probably 10 to 12 carbon atoms. Medium chain, anything between 12 to 16. And long chain is anything longer than 16 carbon atoms. So again, we do have reserves of both carbohydrate and lipid in our body, but very limited reserve of carbohydrate. The reserve of adipose tissue is not only much longer, but it's fairly unlimited. You know, you can have no sort of limited capacity to the amount of adipose tissue that your body can store. In general, actually, most people will probably have anywhere from a 2 to 3 month supply based just on the adipose tissue. And again, that's of course assuming that you're consuming water and any of the micronutrients that you might need to perform enzyme activity. The reason it's nature's favorite storage fuel is it's worth so much, so 9 calories per gram. We're going to talk about several of these as we go through the chapter here, but why fat reserves are important. Hibernating animals are going to use those fat reserves to generate heat by non-shivering thermogenesis. So we'll talk about this in the context of uncoupling proteins. It's really important as an efficient fuel source for migratory birds, and even the hydration of camels. So that hump that's in a camel is actually a large fat deposit, and when you hydrolyze and mobilize those fats, you actually generate water in that process. So one thing that I've kind of put in the middle of these two is ketone bodies. And the reason it's put in the middle is it is a fuel that is derived from fat. So it kind of lives sort of over in this camp, but it is a hydrophilic fuel. So in that way, it sort of mimics the ability of glucose to serve as a hydrophilic circulating fuel. So again, we'll talk a little bit more about ketone bodies as we talk about how they're synthesized, but at the end of the day, you're able to take triglycerides that are stored in adipose tissue, break them down ultimately to acetyl-CoA, and rebuild those into ketone bodies, which you can then circulate throughout the bloodstream. So as an overview of lipid metabolism, at the end of the day, once we get to fatty acids or triglycerides that are found intracellularly, there's no real differentiation whether it came from a dietary consumed lipid source or it was endogenous from adipose tissue. So we do need to talk a little bit about how we get each of these into cells, but once we get in there, then the breakdown process is going to be the same. So let me just first talk a little bit about and highlight the overviews of sort of what happens intracellularly to fatty acids and triglycerides. We are going to study the breakdown process of fatty acids, which is called beta oxidation. So the net outcome of beta oxidation is to produce acetyl-CoA. And again, when we have acetyl-CoA that we've broken down because we're in a need state, we can actually use that locally by breaking it down in the citric acid cycle and then electron transport and oxidative phosphorylation to generate the ATP and the currency that we need. Alternatively, if, again, this is largely in the liver that this is taking place, if you are in a global need state, acetyl-CoA that's broken down in the liver from triglycerides can be made into ketone bodies. And then again, those ketone bodies can be circulated to peripheral tissues. Just highlighting here that, acetyl-CoA is an important branch point metabolite. So it can be made from carbohydrates as we've learned. And in our next learning unit, we'll understand how some amino acids can be converted into acetyl-CoA. Nonetheless, again, excess acetyl-CoA is going to be a different process here. So again, if we've got excess acetyl-CoA, we're in a fed state. That's kind of what we have shown here in green. In that case, we need to be activating fatty acid synthesis. Okay, so that's sort of a fed state pathway. Excess acetyl-CoA can be then made into fatty acids and then assembled on a glycerol backbone to make triglycerides. And actually then those will be put back into adipose tissue as storage. So again, in a sort of very simplified overview, these are the two main pathways that we're going to be talking about in this chapter: the breakdown of fatty acids in a need state and the synthesis of fatty acids in a fed state. Okay, so again, just highlighting, and these were some in blue here. So these were some of the questions and the concepts that were important for your fundamentals sort of questions on your homework, and will be part of your fundamentals quiz that you'll have on lipid metabolism. So when we think about beta oxidation, just going a little bit more into the weeds here, 4 steps occurring in the mitochondria. We define it as aerobic because you do not commit biochemistry to really happen to a significant extent in the mitochondria unless you are in a state of sufficient oxygen. So here's the main equation that I want you to know for beta oxidation. And we've got these written in a specific order so we can sort of couple things together. We're going to have fatty acids that are ultimately broken down into acetyl-CoA. That is an oxidation process that involves carbon-carbon bond cleavage. If we have an oxidation that happens, it must be coupled with a reduction. And reduction, as we see it in beta oxidation, is going to involve both FAD being reduced to FADH2 and then NAD plus being reduced to NADH. So kind of remembering that in that process is a way to kind of keep track of all of the pieces that we have here in our beta oxidation chemistry. So the rate determining step for beta oxidation is not really a specific enzyme, but if we are allowing fatty acids to get into the mitochondria, and as we'll learn later, it's as fatty acid CoA molecules, if we have those getting into the mitochondria, they are going to be broken down by this beta oxidation process. Coming over here to the flip side and thinking about fatty acid synthesis, this is actually a 7-step process that occurs on a single enzyme. It occurs in the cytosol by the enzyme fatty acid synthase. It's technically an anaerobic process. We don't need to have oxygen around for this process to occur. And here's the net chemistry for what we have here. A little bit of a longer reaction. So I've got the 4 species up on top here that are going to be responsible for sort of initiating. There are reactants for this process. And then again, here are the products. So let's couple these together in this specific order so that we can help sort of remember these steps. So in this case, we're going to have acetyl-CoA and then bicarbonate as a carbon source being involved in the synthesis of fatty acids and then liberating CO2. As we'll learn in this process, the whole purpose of this is not necessarily to generate CO2 as a waste product, but what we really have here is one acetyl-CoA molecule is going to be our electrophile, and then by adding a carbon atom and generating a beta-keto acid, we are generating an important nucleophile that allows us to net couple 2 acetyl-CoAs together and make our fatty acids. So again, not just 2 acetyl-CoAs, but couple acetyl-CoAs together, carbon-carbon bond forming chemistry. This is going to be reductive chemistry because remember, our acetyl-CoA has that carbonyl functional group. Fatty acids are largely hydrocarbons. So we do have to do some reduction chemistry. So these guys get coupled together because they're related to the chemistry that happens with fatty acid synthase. We need some glue to do this chemistry together. We need some reductive currency. So NADPH is our reductive biocurrency that we're going to use here. And again, that's oxidation, taking NAD plus, taking its electrons and generating NADP plus. And so that is, again, going to allow us to do that reductive biosynthesis, coupling acetyl-CoA together, using some of those electrons as glue for that carbon-carbon bond forming chemistry to generate our fatty acids. This process is not without an energy requirement, so we utilize some ATP for that. So that's the purpose of having ATP in this chemistry. And again, the product of using ATP as an energy source is to have ADP as a product. Rate determining step here for fatty acid synthesis is ACC, acetyl-CoA carboxylase. So actually none of these 7 steps that we have here in fatty acid synthase are really rate controlling. The rate controlling step is actually by acetyl-CoA carboxylase, which is involved in this chemistry that we have here. So a lot there sort of on that slide, but what we're going to focus on the next little bit here is talking about how we get dietary sources of triglycerides, ultimately as intracellular fatty acids to do this biochemistry. And just to highlight here, about 90% of the dietary fats that are consumed are triglycerides or cholesterol. There's very little that's other molecules. But remember, when we talked about other lipid molecules in Chapter 9, we had things like our fat-soluble vitamins and so forth. But again, most of the things that are labeled as dietary fats are really triglycerides and cholesterol. All right, so this is going to be an important overarching picture that we're going to kind of look at over the next few slides here. So again, what we're highlighting here is some dietary fat that we've consumed. So this is exogenous kind of fat here. We're going to talk about what happens as you go through the stomach, through the small intestine, and then allowing those digested and hydrolyzed species to pass through the intestinal epithelial wall, ultimately getting first into the intestinal lymph and ultimately into the bloodstream. Once we get these molecules into the bloodstream, they stop at the liver, often get repackaged, and then recirculated out to peripheral tissues. So again, that portal vein allows for an initial stop here at a liver to do sort of like a central processing sort of facility. So we've got liver cells that we'll kind of consider what's happening with here, our adipose tissue, and our muscle tissue. The only thing I don't like about this figure is it kind of implies that there's a one-way direction, kind of getting these molecules into adipose tissue and muscle cells. And where we really don't have an out pathway with muscle cells, that is a really important component of adipose tissue. Yes, when we have excess, we store, but it's just as important that we mobilize fuels from adipose tissue. But again, if we're mainly talking about exogenous lipid pathways, adipose tissue really represents an endogenous source. So I'm certain that's why we don't see that kind of highlighted on this picture. All right, so again, we're just going to go through and we've sort of highlighted on this little mini version of this picture where we are at when we're talking about some specific processes here. So we're going to start with the stomach. And again, dietary lipids are largely hydrophobic and your digestive environment is largely hydrophilic. And specifically, as we're talking about the stomach, we've got a very low pH. So if we've got to think about metabolizing foods that we've consumed, we need to think about how we connect and interact the enzymes with the food sources or whatever reagents we have that are going to help us digest these materials. So there's three things that sort of help with this process. The first is just the physical mixing that happens with the peristalsis in your intestines. And again, the purpose of that mixing is to increase the surface area that lipids and water and the enzymes contained in that aqueous environment will be able to interact under. So again, that physical mixing is very important. We also talk about this concept of emulsification. So again, we increase surface area by mixing, but we also can sort of keep that mixed solution emulsified or kind of keep it from reseparating back out into water and oil layers. And we do that by using a soap molecule that really is sort of nature's soap, and that's bile. So what we have shown here on the right is a bile acid, and hopefully you kind of look at the structure and you say, oh, that looks like a steroid. That looks very similar to what I think about for cholesterol, and that's absolutely true, and that's because bile acids are actually derived from cholesterol, and cholesterol in excess is actually eliminated by its production into bile acid, which is excreted then into your digestive tract and allowed for its elimination. So again, bile is synthesized in the liver, it's stored in your gallbladder, and again, just to highlight again, it's synthesized from cholesterol. So the only real route that your body has for ridding itself of excess cholesterol is its production into bile acids and its excretion into your digestive tract. The last thing to kind of highlight here when we think about things that start happening in your stomach is we have these phospholipase enzymes. Remember, we've got that ester linkage in triglycerides that we're going to need to hydrolyze. Let me flip back here just for a second to highlight, right? This is our ester linkage that we have right here, right? We've got our fatty acids that are esterified on a glycerol backbone. So we need to hydrolyze this ester linkage there. And we've got enzymes that can help do that. So we have these phospholipase enzymes that are going to do ester hydrolysis. So again, one of the first mechanisms that was part of your homework here is homework question number 3, and that's drawing a mechanism to show the hydrolysis of an ester linkage in acidic solution to generate free fatty acids and glycerol. Now again, we're drawing a very generic reaction mechanism here. We're not specifically drawing it with fatty acids, but we can kind of map on what we think about here. So again, we've got our generic, and I'm not going to go through a lot of details here because there already is a video up kind of talking about and walking through this mechanism. But if we were to map on what we see here, if this was a glycerol backbone, right? So we would have our ester linkage here. So here's our ester linkage. And then so this portion here, we could imagine this is going to be our fatty acid. And then this portion right here is going to be part of our glycerol backbone. So we'd actually have 3 of these together here connected to make our glycerol backbone. So again, I'm not going to go through the details of this mechanism on this video. There was already a video that was posted that kind of went through all 4 of the mechanisms that we have from this chapter. Okay, so now we're into the small intestine. And we don't really have this highlighted here, but again, here is the small intestine. So we've gotten past the stomach, we've emulsified, we've done some mixing with peristalsis. And so a big theme that I want to make sure we remember is that only monomers are going to be able to exit the gastrointestinal tract. So that was part of kind of a homework question there, to remind ourselves that only monomers exit the GI tract. If you have things that are larger than monomers that are exiting your GI tract through the appropriate channels, it's likely that you have leaky gut. And again, that can be a problem that can lead to significant immunocompromised sort of situation. So kind of reminding ourselves of the 3 main macromolecules that we've focused our study on. We've got carbohydrates, we've got lipids and proteins. Only really 2 are considered primarily to be fuel: carbohydrates and lipids. So we'll learn in our next learning unit that yes, proteins can be used as a fuel, but their main purpose is to be building blocks, not only to make other proteins, but to make neurotransmitters, to make cell signaling molecules, lots of other important purposes for amino acids. But again, they're meant to be building blocks, not necessarily fuel. So I've got a few little stories here to talk about each of these. I'm going to talk about the non-lipid stories first, and then we'll wrap up kind of with the lipid story that's sort of relevant here. So some of this stuff, as we talk about some things with carbohydrates and proteins, should be review from some of the things that we talked about last semester. So we've got carbohydrates here, right, that can be broken down into their monomeric form as monosaccharides. And this is important to highlight, right? You can't see, you will never see disaccharides like sucrose in blood. And I think the little story that we told in class was highlighting that that's a way that you can sort of tell if you've got sort of gastrointestinal perforation. I read a study that they were looking at it with horses, that they could tell if there was some gastrointestinal sort of perforation if you saw sucrose in the blood, because you shouldn't see sucrose in the blood because it should not be getting out of the digestive tract because it's not a monosaccharide. Same thing with fiber, right? That's a very large polysaccharide. But again, we talked just a little bit here in class, reminding ourselves that things that are fiber have beta 1,4 linkages. And those are really important because not only do they provide bulk to help move intestinal waste, but often these fibers are fuel for your microbiota. So the food part is what we refer to as prebiotics. The microbiota are what are the probiotics. So these are the bugs that live in your belly, and these are the food that they eat. So you need to feed your probiotics prebiotics. And again, just highlighting that anything that makes its way down into your gastrointestinal tract, specifically, again, your colon, anytime we're talking about this microbiome, that's really talking about your large intestine. So even here that we're talking about small intestine, your microbiome really lives exclusively in your large intestine. It's not supposed to be in your small intestine. And we've talked about that concept of SIBO, or small intestinal bacterial overgrowth. When you actually have microbiota from your large intestine that make their way up into your small intestine, that's a problem. But again, if you've got either food that gets down in an indigestible format into your large intestine, that can cause gas and bloating because sometimes your probiotic bacteria begin to break that down. We talked a little bit about sucralose, right, being an artificial sweetener. Sucralose, however, if it makes its way down into your lower GI tract, there are some studies that suggest it might have some toxicity to the bacteria that are supposed to be living healthfully in your large intestine. All right, a little bit of a story here now about amino acids. And one of the amino acid stories, again, we're connecting all of these together because we're talking about food additives here. But aspartame is actually a small little dipeptide. It's made by coupling phenylalanine with aspartic acid, and rather than having a carboxylate group there, we actually create a methyl ester. So we create a methyl ester there. And two reasons why aspartame is thought to be problematic is it is a little peptide, so you will hydrolyze it, and that means you make free phenylalanine, and the concern is that you could potentially make free methanol. So depending on how much you consume, the thought is that could be problematic. This is something that we'll get to in our next learning unit when we talk about protein and amino acid metabolism. There are some individuals who have problems digesting phenylalanine, and problems digesting phenylalanine are individuals who have a metabolic disorder called PKU, or phenylketonuria, which means they have elevated levels of phenylalanine in their bloodstream. So we'll talk about sort of the implications of that as we get into our next learning unit. All right, so let's then talk about fats. Again, fats, we need to think about getting fatty acids out of your digestive tract. And so even though you've consumed these triglycerides, remember earlier on we had those phospholipases that would break them down into their free fatty acids. That continues to happen in the small intestine. And again, we've got to take and get these fatty acids out of your intestinal tract and ultimately into your bloodstream. But again, this is problematic because these are largely insoluble. So this is going to be a theme that we'll see here throughout this chapter, is anything that is a lipid often has to be complexed with a protein in order to be transported. So just as a little highlight, anytime we're talking about and seeing these fatty acids, this is going to be these little black sticks that we see here. So on this little figure, any of those little black sticks are free fatty acids. So when we want to get fatty acids out of our intestinal tract and into our intestinal epithelial cells, we need to complex them with a fatty acid binding protein. Okay, so fatty acid binding protein is kind of shown in a structural form here. It's a very small protein, not a lot to it. It's what we call a beta barrel protein, a little bit of alpha helical character here. But here you can see the fatty acid is literally kind of sequestered and housed within this fatty acid binding protein. So the outside of this fatty acid binding protein is very water soluble. So almost like a little micelle, you pull that fatty acid inside and you sort of disguise it. So a homework question here kind of relating to how and why this works. It's really important because it really increases the solubility of fatty acids. Again, that's a theme that we're going to see. You're going to complex lipids with proteins to make them more soluble. And this is also important because it helps with the facilitated diffusion and the influx of fatty acids into your intestinal epithelial cells. Because once we get inside our intestinal epithelial cells here and we complex with this fatty acid binding protein, it looks like there's no fatty acids on the inside here. So from a facilitated diffusion standpoint, you're going to continue to pull those fatty acids into your intestinal epithelial cells. All right, last little story here to sort of talk about a food additive sort of story with lipids is to talk about olestra, which is also called olein. So if we think about, remember the structure of triglycerides, we've got a glycerol backbone and then we've got three fatty acids that are appended to that. What we have with olestra is we essentially take a sugar that has a whole bunch of hydroxyl groups, kind of like glycerol has hydroxyl groups, and we're going to attach fatty acids onto it by making an ester linkage. So rather than having, and let me flip back here for a second, rather than having a glycerol backbone here that has three fatty acids appended on it, you essentially use something like glucose and you're going to hook fatty acids onto any of the hydroxyl groups that you have there. So it's almost like a wheel, if you will, of fatty acids. Now, why that's problematic is we do not have enzyme machinery that can break that ester linkage. Our phospholipases don't act on that. Okay? Well, why this is important as a fat additive or a fat substitute is it has the taste and the texture of real fat. Okay, so when you consume it and it looks like this on your taste buds, it has the taste and texture of fat, so that's important. But the problem is it gets down into your digestive tract and your lower intestines having not been metabolized at all. So basically, you get down into your lower GI tract and you have an oil that's not been broken down, not uptaken into your bloodstream. And again, that's why it's fat-free, because it doesn't get taken up. Now the problem is it exits out in the same form that it entered. So you'll see warning labels on anything that contains olestra or olein to say warning may cause oily, runny stool. And that's because you have no mechanism to sort of break down this fat oil substance as it goes through the entirety of your digestive tract. All right, so once we get things into the bloodstream, now we need to think about how we're going to package and complex things. So again, we are down sort of here and talking about not only getting into the bloodstream, but first sort of into the lymph. So one of the things that I want to talk about is these little blue spheres that we have here that are called chylomicrons. So these chylomicrons are what we have over here in our little image that we have here down at the bottom. Chylomicrons you can sort of think about as being like big fat buses. So the yellow and the blue that we have that are passengers, if you will, on these buses represent triglycerides. So we're going to have triglycerides here in yellow. And then we're going to have cholesterol esters. So once we look at the structure of cholesterol, but if you can envision this in your head, cholesterol has, like the OL name or a suffix at the end of it, it has an alcohol functional group. So like glycerol has an alcohol functional group, and like sugars have an alcohol functional group that we just talked about making olestra, cholesterol has an alcohol functional group that we can hook a fatty acid to. So these cholesterol esters really are cholesterol that has a fatty acid hooked to it. So that's what these little blue guys are. So all of the things, all of the passengers that we have in chylomicrons are basically from dietary lipids. So dietary triglycerides and dietary cholesterol that you're going to pack into these chylomicrons. So these chylomicrons are what exit out of your intestinal epithelial cells. And what we'll see here is this is, again, a giant vehicle. We can see a lipid bilayer that we have here. These green things represent proteins that we have on the surface of this transporter, and that's going to direct this particle to its destination. And so one of the things that we're going to see here is we're going to notice a couple of these receptors that we're going to have here that are going to indicate where these guys go. If you have this A1 protein, so this is an apolipoprotein A1, if you have that, that literally is a ticket that tells you to go to the liver. So what we'll see is we've got HDL particles in chylomicrons that have directions that allow them to go to the liver. So again, if we follow the pathway that we have here, chylomicrons again have dietary triglycerides and cholesterol. They go into the intestinal lymph, go into the bloodstream, and through the portal vein, first are allowed to sort of offload some fatty acids at some initial tissues if they need, but ultimately we're going to have these cholesterol-rich chylomicron remnants that end up going to the liver here. So what we can see here, we've got these blue blobs that are kind of big. They've got lots of these fatty acids and triglycerides that are in here. We're going to allow some of these to be delivered to either muscle cells or adipose tissue. But at the end of the day, once they offload some of these yellow passengers, we're going to be cholesterol rich, because we're going to have a lot of these blue passengers left. And these are cholesterol rich chylomicron remnants. So again, these are going to end up being taken up by the liver. So that's sort of the pathway that we have here. We've got this chylomicron remnants that are very cholesterol rich. These are going to be taken up by the liver through a process called receptor-mediated endocytosis. We're going to get to that in a couple of slides here. But you'll notice that HDL particles can be taken up by the same process because they have the same sort of protein receptor. So just to highlight here, if it wasn't clear, all of these represent what we call lipoproteins. So they've got lipids. They've got lipids as the lipid bilayer here in these micelles. They're filled with lipids, but then they have proteins that are wrapped around them. And the proteins that are wrapped around them, just like we saw with fatty acid binding protein, proteins can have nice hydrophilic exteriors that allow them to be water soluble in the bloodstream. But you can have the inside be filled with fat. And the same is true here. We've got our hydrophilic head groups on our lipid bilayer here, or I should say our micelle. And then we also have some proteins here that not only increase the water solubility, but also provide directions for where these particles are supposed to go. But the inside of all of these is always going to be filled with lipids. And it's going to be some combination of triglycerides and then cholesterol esters. So again, we've got then these in question 7 in your homework talking about these cholesterol remnants are then, or these chylomicron remnants are picked up by the liver by receptor-mediated endocytosis. They're going to be repackaged with triglycerides and cholesterol that are made from your own body in your liver. So again, inside here, and I know this is a small picture to see here, let's kind of blow this up. When we've got these cholesterol-rich chylomicron remnants uptaken by the liver by receptor-mediated endocytosis, we're going to have now this dietary cholesterol that's left here be repackaged with cholesterol that maybe you've made yourself and or triglycerides that you've made yourself in your liver. And then these are going to be offloaded and returned back out to the bloodstream. So one of the things that I want to highlight is once you become a VLDL particle, not only do you sort of change the composition of what's on the inside, but very importantly, you change your directions on the outside. So chylomicron remnants don't necessarily just become VLDL particles. You really sort of exchange your contents and fill them into a new vehicle. So the new vehicle, which is a VLDL particle, is going to have this ApoB100 protein. Okay, so I do want you to know the names of these different proteins. So apolipoprotein A1 is what's going to direct things to the liver. B100 is what's going to direct things away from the liver and to peripheral tissues. So VLDL particle stands for very low density lipoprotein, and that's because they're very full of lipids on the inside. So they're low density. Lipids are less dense than proteins, and so this is a very low density. As these guys begin to deliver their yellow contents here, their triglycerides to peripheral tissues, they become LDL particles, which again, now are only really full of cholesterol. So again, we can sort of see this here if we look at our picture here. We've got these VLDL particles that are going to circulate throughout the bloodstream. They're going to be offloading their fatty acids to destinations, whether they're for storage in adipose tissue, use in muscle tissue. And again, they become these LDL particles because they've offloaded all of their components. The last little thing I want to make sure to highlight on this slide is another protein that's important for packaging fatty acids. So remember how over here we talked about the importance of fatty acids being complex with that fatty acid binding protein. Even though they show these little black sticks here just kind of floating around, we're never going to have lipids just floating around like that by themselves kind of in your bloodstream. They need to be complexed with a protein. And fatty acids complex with the protein albumin in your bloodstream. So another protein that I want you to know there. All right, so now once we talk about finally getting to our delivered tissues, we're going to focus largely on muscle versus adipose tissue. Well, things are going to get delivered to your muscle tissue if you're in a need state because your muscle can use those fatty acids as fuel. If nobody is picking up those fatty acids because nobody needs energy, then they're going to be delivered to adipose tissue. Again, we can think about this as being a fed state tissue, and then they're going to be stored in your adipose tissue. So again, whether we uptake these fatty acid albumin particles or LDL particles for that matter by muscle or adipose tissue sort of depends on the state. Again, if we are taking things up by things like muscles and we're in a need state, we're going to use those fatty acids for energy. And again, that's going to be that beta oxidation process. If we are in a fed state and we're delivering those fatty acids to adipose tissue, that means we're going to be storing them as triglycerides. So an important piece to this puzzle is really the ability to tap into, well, what if you're in a need state, and what if you actually don't have some fatty acids that are kind of circulating around in your bloodstream? You need to be able to mobilize the ones that are stored in your adipose tissue to be able to use for energy. So this is a really important sort of component to be able to tap into. This is hormonally regulated. We're going to kind of deal with this when we talk about regulation of this pathway. So again, we need to be able to take fatty acids that are stored as triglycerides, convert them into acetyl-CoA, and either use them locally for energy or convert them to ketone bodies if we need to be able to help other tissues. But this is really important to highlight that this is hormonally regulated, because if you have some instances of hormonal imbalance, and we'll revisit insulin at the very end of our course here, you have a really hard time kind of tapping into your storage if you have sort of high levels of insulin and you've become insulin resistant. Okay, so a lot in that first lecture kind of dealing with how we mobilize fatty acids, either by going through the digestive process, or again, we really haven't talked about too much how we sort of deliver or get them from endogenous sources. But again, lots to kind of get us to the point where now we're going to have fatty acids that are inside of a cell.